Hello and welcome. My name is Erica Shugart and I'm the Director of Communications and Marketing Strategy here at the American Society for Microbiology. I'm delighted that all of you could come out through the snow and everything else that's out there to come to the ASM headquarters and join us for this evening. And I'd also like to welcome our internet audience out on live stream, and I was glad that we can have people from around the world join us as well for this fabulous lecture this evening. Our featured speaker tonight is Dr. Martin Blaser. He is the Muriel and George Singer Professor of Medicine, Professor of Microbiology, and the Director of the Human Microbiome Project at New York University School of Medicine. So he's come down here from just up the coast. He went to the University of Pennsylvania for his undergraduate work and then did his medical degree at New York University School of Medicine. He's been studying the role of bacteria in human disease for the past 30 years. His lab focuses on bacteria in the human microbiome, particularly looking at the biology of colonization and the nature of the interactions that can lead to or prevent disease. He also does ongoing work on the focus of the normal microbiome and how it can be perturbed, which is some of what we'll be hearing about this evening. In 2014, he published a book, Missing Microbes, which if you haven't already purchased it out in the lobby, can be purchased afterwards and signed by Dr. Blaser, so I certainly hope you will. It's a fabulous book, very interesting. We're going to hear about that this evening. Dr. Blaser is the past president of the Infectious Disease Society of America. He's a fellow of the American Academy of Microbiology. He was elected to the Institute of Medicine at the National Academy of Sciences in 2011, and in 2015, he was named one of Time Magazine's 100 most influential people. So a lot of accolades, and we are delighted that we're able to have him here. So we're going to have him come up. He's going to tell us about missing microbes, and afterwards we will have a chance for questions. If you're out on the internet and you'd like to ask questions, you can either ask them in the comment box that's directly below the video, or you can tweet them to us at ASM Live. So with that, I'd like to welcome Dr. Martin Blaser.
So thank you, Erica. Thank you for your lovely invitation and your even more lovely introduction. I'm glad that the snowstorm was a few days ago and not tonight, and so that people could actually come here. And it's a pleasure to be here at ASM headquarters because I've been a member of ASM since 1980, before some of the people here were born. And ASM is a great organization. I've been taught by microbes, and ASM is the organization that involves people who want to understand microbes and use that understanding to better human health, human business, human farms, the oceans, everything. So this evening, I'm going to talk about what happens when our early life microbes are perturbed. And I want to start with this slide, which is, it's entitled Views of the Greenland Ice Sheet. And here are two satellite photos of Greenland, 1992 and 2002. And you can see that in one of them, there's a lot more white in the slide. And we know that that's ice. And we take this slide to be a representation of global warming. And we can think about global warming as a change in our macroecology due to human activities. And what I'm going to talk about is a change in our microecology. And I'm going to make the point that the scale of the problem may be equivalent to this scale as well. So let's begin at the beginning. This is a representation of ancient people and their ancient microbes. And what we know is that ever since there were animals on this planet, they have had microbes living in and on them. Every animal that evolved in relationship to the microbes because the microbes came first, certainly the bacteria. We know that the microbes do not colonize their host randomly. They live in specific niches. That means that for us people, the microbes on your arm are different than the microbes in your mouth. Many of the microbes are persistent. You acquire them early in life. They're present for weeks, months, years, decades, for your entire life. Some of the microbes are conserved. What that means is that there are certain microbes that everybody in this room shares, and others are host-specific. And in a room this size, it is fair to say that each of you has microbes that no one else in the room has. So that creates an interesting tension, the tension between the conservation and the host specificity. Now here is a microbiologist's view of the human body. Human cells in red, microbial cells clear or in white. We have more bacterial cells in our body than we have human cells. The exact numbers are not entirely clear, but all the estimates mean that the bacteria outnumber us. It leads to this question: who are we? Here's a geneticist's view of the human body. Human genes, 23,000 in red. Microbial genes, unique microbial genes, an average of about 2 million. So many more microbial genes than human genes. And we might ask from that, what are all these genes doing? In fact, we can ask a series of biological questions about the human microbiome. What's the identity of the microbes that populate their host? What are they doing? How is the host responding to them? What are the forces that maintain equilibrium across the populations? Why don't they kill us? Why don't we kill them? And what are the unique characteristics of each individual? And very importantly, how can we manipulate it to improve health and prevent disease? So one of the questions that comes up is exactly when does the microbiome become established? And when does the microbiome of us adults become established? And this was looked at in a very nice study published in 2012 in Nature by Yatsenenko and colleagues. This is from Jeff Gordon's lab. It involves the work with Rob Knight and Maria Gloria Dominguez, among others. It's a study of people in 3 countries: people in the US, Amerindians in South America, and Malawians in Africa. And in this graph, each circle is the composite of the gut microbiome in one individual. The x-axis is age during childhood, and the y-axis is something called the unifrac distance between children and adults. And this is actually an index of dissimilarity. And what the data show is that when babies are born, when they're young infants, their microbiome is very unadult-like. And then gradually and progressively, it becomes more and more adult-like until it is. And what's remarkable here is the age of transition is the age of 3 years. So the first 3 years of life is when the microbiome is developing. It's the most dynamic. It's also the period of life when babies are developing. They're developing their immunity, their metabolism, their cognition. Now, this slide is a schematic of a hypothesis about the interaction between co-evolved colonizing microbes and their host. This is from work that Denise Kirshner and I did. Denise is a mathematician at the University of Michigan. Our concept of coevolution are microbes that know how to talk to the host. They provide signals to the host, and the host signals them back. These could be chemical signals, physical signals. And in our model, it forms a kind of network, an equilibrium. Denise has shown that this kind of model is quite robust and resilient. And what I want to discuss is what happens when these ancient relationships become perturbed. What happens when they're lost? Now, this slide examines the evolutionary relationships of wild hominids. This is work by Howard Achman and his colleagues. Here on the left side is a phylogeny of wild hominids. Here's one, for example. And this phylogeny has a certain topology. There's a branching pattern. And this is based on the host genes, on the host DNA. And on the right side, this panel is the phylogeny based on their fecal microbes, the microbes in their intestinal tract. And if you look at the colors and you look at the branching pattern, you'll see that it's almost the same as what we see in their host phylogeny. So this congruence is strong evidence for vertical transmission of the microbiota, and also it's evidence to support this idea of coevolution. The time frame of this phylogeny is about 7 or 8 million years. That's how long we were together. So we have evidence that we co-evolved with our microbes for at least 7 to 8 million years. We have evidence probably that it's since we became mammals, maybe 100 million years. So it's a very ancient kind of relationship. And how does it start? Well, this slide looks at the transfer of microbes from mothers to children, what I call ancient, because we humans are mammals. And the nature of being a mammal is that we're all born in a womb. It is sterile or mostly sterile. And our big introduction to microbes occurs at the moment that the water breaks. And the bacteria in the mom's vagina ascend. And as the baby descends through the birth canal, the baby is covered by bacteria from the mother. The baby swallows bacteria from the mother. That's their first inoculum of bacteria. The baby has skin-to-skin contact with the mother. The baby's mouth filled with bacteria inoculate the breast. And now these bacteria go in with their substrate, milk, which has substrates that are specific for the microbiota. And that becomes the foundation of the gastrointestinal tract microbiota. Moms are kissing and licking babies. They're pre-masticating food. Nature has a lot of very efficient ways of transferring microbes from moms to babies. And this is what's happened in all mammals. And there's a fair amount of redundancy here. So this is the intergenerational transfer of microbes. But these days, moms aren't the same as they used to be. In this modern world, moms are in an environment with antisepsis. They've received many courses of antibiotics, including when they're pregnant, when they're about to deliver. And babies aren't the same. For example, babies may be born by cesarean section. Now in the United States, about 32% of all births are by C-section. 1 baby out of 3 is born by C-section. In some countries, it's 1 baby out of 2. In some cities, it's 8 babies out of 10. In Rio de Janeiro, in Rome, even in Shanghai, it's more than 50% of babies born by C-section. This is a big change in human biology. Babies are bathed extensively from the moment of birth. Many of the nutrients and compounds in vernix are removed. Babies are getting formula at a very high rate. Formula superficially resembles human milk. It's a white liquid. That's about it. And of course, babies get a lot of antibiotics, and I'm going to discuss that. So based on these kinds of differences, over the last about 2 decades, I've been formulating an idea that I call the disappearing microbiota hypothesis. And it has 4 main tenets. First, beginning in the late 19th century, changing human ecology has dramatically altered the transmission and maintenance of our indigenous microbiota. These changes have affected its composition. We have evidence for this. This altered composition affects human physiology and thus disease risk. We have evidence for this with Helicobacter in the stomach. And loss of ancestral bacteria, usually acquired early in life, is especially important because it affects a developmentally critical stage. This is the hypothesis. About 6 years ago, I extended the hypothesis working with Dr. Stanley Falco at Stanford. And the extended hypothesis is shown here, the effect of maternal status on the resident microbiota of the next generation. Our idea is that ancient moms had an ancient microbiota. And if they happened to lose microbiota, they might be able to get it back horizontally. But if they couldn't, then the next generation would be born with a less diverse microbiota, and so on and so forth. And in this model, our microbiota has been stepping down over generations. So if this is true, this might be very important. Is it true? Well, we're already beginning to find evidence that it is true. Here's a study from Ureta and colleagues looking at Helicobacter pylori, the dominant organism in the gastric microbiota, in 3 generations in Japan, grandmothers, mothers, children. Looks a little like the theoretical step. Now I'd like to tell you about some work that was published not long ago, some studies of isolated Amerindians in a village that was cut off, that never had contact with Westerners before 2008. The village was spotted in the air by a military helicopter in the remote areas of the high Orinoco in Venezuela. The people were Yanomamis. A Yanomami interpreter came down and told the people that they were with doctors. The village they found looked like this. This was the village. The people were hunter-gatherers. They had 2 metal objects in the village, a machete and a can, but otherwise were in the Stone Age. They had no written language. But they had heard of medicinas. They had traded with other Yanomamis. And so a team of doctors went in to visit this village to vaccinate them, because once a village is spotted, they're at risk for infectious diseases. And the policy of the Venezuelan government is to vaccinate them. And a study was done, conducted by Maria Gloria Dominguez, who's also my wife. And here we're looking at the amount of diversity in people in the US and people in 2 villages in Africa and in South America that have had some modernization. And here are the Yanomami. And if you look at it the other way, they have the most diversity, and then it has stepped down in these other populations. This also is consistent with the hypothesis. And if you compare the amount of diversity in the Yanomami and the people in the US, it suggests that we've lost about half of our diversity already. So the question is, firstly, is it true? And second, if it's true, what are the consequences? Is this just an academic point, or could there be consequences? So I now want to turn to a problem that all of you are aware of, and that is obesity. This slide shows obesity maps in the United States from the CDC, 1989 to 2010. And in 1989, there's no state in the United States where more than 14% of adults are obese. By 2010, there's no state with less than 20% obesity, and the national average is 30%. What's remarkable is that it is happening all over the United States, and the difference between the first map and the second map is only 21 years. Something very powerful must be going on. It's not just true in the United States. It's true in many countries of the world, maybe in all countries of the world. Here's a map from Netherlands, 1981, 2011. In the 30-year period, you can see that the proportion of overweight adults has increased dramatically. Here are more data looking at populations. Here are obesity trends among US children and adolescents. These are data from NHANES over about a 35-year period, looking at children as young as 2 to 5 years old. And you can see that in every age group, the rates of obesity have gone up. And this slide is representative of a wide body of data that the seeds for obesity are beginning early in childhood. So what could be causing this? We know that calories are important, either how we burn them or what we ingest. But I'm going to make the hypothesis that one of the great miracles of 20th century science, the discovery of antibiotics and its use, has something to do with this. I show this picture of Alexander Fleming and his famous observation of the zone of inhibition around a penicillium mold inhibiting Staphylococcus. And when penicillin and other antibiotics came on the scene in the 1940s, they were miraculous. They saved lives of people who had what would have otherwise been fatal illness. And the public and the health professions alike came to the view that antibiotics were miraculous. And as a result, antibiotics began being used more and more and more. So how much more? So here's a recent study of more than 1 million pregnant women in the United States. And of these women, 82% of them received some drug during pregnancy. And of the top 5 drugs, 4 of them are antibiotics. We estimate 50 to 60% of American women are getting an antibiotic during pregnancy. How about childhood? Here are the top 8 prescriptions in US children in 2010. And of the top 8, 5 of them are antibiotics. These 5 antibiotics alone were given more than 40 million times that year. The other 3, interestingly, are for asthma. Antibiotics are everywhere where our modern society has gone because people understand the power of antibiotics and they want to get the benefit of the antibiotics. This slide is from a photograph I took on my phone in a tiny little village in Amazonian Peru. There, between the drinks and the batteries, were the over-the-counter antibiotics, generic, all made in China. It's very hard to find places where people haven't been on antibiotics unless you go to uncontacted villages. How about the totality of antibiotic use in the United States? So here are more data from the CDC. Outpatient antibiotic use in 2010. And the bottom line is that there were 258 million courses of antibiotics prescribed in that year. And that's a rate of 833 courses per 1,000 population. That's 5 courses for every 6 people. And this has been going on year after year. The highest rate is in young children. It then goes down in young adulthood and then comes back up as people get older. And because of the stability of use, one can estimate the total exposure. And the estimates suggest that in the first 2 years of life, the average child in the US has received nearly 3 courses of antibiotics. And by the time they're 10 courses. And by the time they're 20, 17 courses. These are big numbers, but they're consistent with many other studies, albeit those were on a smaller scale, showing this. The data from 2011 look about the same, except slightly worse. Now, the CDC investigators, and this is Laurie Hicks and colleagues, looked at the geography of antibiotic use. The national rate was 833 per 1,000, Northeast 830, Midwest 868, West 638, much lower than the national average. South 936, much higher. 50% difference between South and West. No 50% difference in the rate of serious bacterial infections. These differences reflect differences in practice and culture, and wherever you look, you find variation in use of antibiotics by region, by practice, by country. Now, it's interesting to look at these 2 CDC maps side by side, both 2010. On the left, obesity, on the right, antibiotics. You can see that there's a lot of resemblance between these 2 maps, and 2011 map just about the same. So these are observational data. There's correlation here. But we don't know anything about causality. We don't know if A causes B causes A, or C might cause A and B. But they get our attention. Now here are some good-looking cows. And so the question is, do antibiotics have any role in metabolism and development? And so I want to bring us back to the farm, where for 70 years farmers learned that feeding low doses of antibiotics to farm animals would promote their growth. And it's so widely done because it works. And this creates a huge problem in human resistance. I'm happy that David Vicey is here. He works with Congresswoman Louise Slaughter, who has been the leader on Capitol Hill to try to eliminate antibiotic use on the farm because of the problem of resistance. And yet, it teaches us something fundamental about biology. We know that farmers can give subtherapeutic antibiotic treatments, which we call STAT, for the growth promotion of livestock. It works between chickens and cows, wide swath of vertebrate evolution. It works with virtually every antibacterial used, regardless of its chemical structure, class, target, or spectrum. Antifungals don't work, antivirals don't work. Farmers discovered that the earlier in life they start the antibiotics, the bigger the effect on growth rate and on feed efficiency, the ability to convert food calories into body mass. That's what farmers want to do. And when I saw these slides, it suggested to me that we're looking at a developmental phenomenon. So we began to do studies in mice in which we would give mice antibiotics or not. We'd examine phenotypes, we'd look at the microbiome, and we'd look for relationships. Now I want to show you several experiments where we've tested these hypotheses. The first experiment was started by Dr. Il-Sung Cho when he was a postdoctoral fellow in my lab. He's now on the faculty of gastroenterology at NYU. Il-Sung took 4 different regimens approved on the farm. He took the midpoint of the FDA approved levels, and here we're looking at percent body fat of control mice, no antibiotics, or mice that received antibiotics. And this was the first result in our hands that showed that antibiotics would change the development of mice. They changed body composition. Il-Sung's study showed many other things, but I don't have time to go much further. I want to go to the next study, which was done by Laurie Cox when she was a graduate student in my lab. Laurie did an experiment that we called FATSTAT to examine what are the effects of combining a high-fat diet with the STAT antibiotic on body composition. So mice received either low-dose penicillin or not. They were all started on normal chow, and then at week 17 of age, half the mice were put on a high-fat diet, and half the mice remained on the normal chow. So now we're going to look at the results in terms of the morphometry of mice, male mice and female mice, total mass, fat mass, and lean mass. So if we start with the male mice, total mass, black line is the control group, normal chow, no antibiotics. On antibiotics, the mice got bigger, just like on the farm. On high-fat diet, they got bigger still. And on high-fat plus antibiotics, they got big-est. Lean mass, increased muscle mass in the mice on antibiotics, just like on the farm. Fat mass, big increase in fat in mice on the high-fat diet, even more when they're on high-fat plus antibiotics. Female mice, many of the same findings. Let's look specifically at fat mass. The average female mouse on the high-fat diet gained 5 grams of body fat. High-fat diet plus antibiotics, 10 grams. They doubled the amount of body fat. The antibiotic potentiated the effect of the high-fat diet. Obviously, you can see that life is unfair, that females had a bigger effect than males. So up to this point, the mice had gotten antibiotics all their life. So Laurie did an experiment that we called Durastat, asking, are the changes in body phenotype durable with just a limited antibiotic exposure? So in addition to no antibiotics and lifetime antibiotics, there were mice that only got 8 weeks or only 4 weeks of antibiotics. Here we're looking at total mass, lean mass, and fat mass. The black line is the control group. We can see that in all cases, all the antibiotic courses increased total mass, lean mass, and fat mass. So 4 weeks early in life was sufficient. They didn't need the lifetime exposure. What about the microbiome? So this slide is a principal coordinates analysis of population structure of the microbiota in these mice at 3 weeks of age. And at 3 weeks, there are only 2 groups of mice: control mice, no antibiotics, or all 3 groups of mice at 3 weeks are still receiving the antibiotics. So in this principal coordinates analysis, each circle represents the composite of the gut microbiota in one mouse. Black is control, orange is stat. Here are the controls in black. Here's the antibiotic group in orange. The distributions are similar, but they're not identical. That's not surprising. One group was receiving antibiotics, the other wasn't. Now let's go to 8 weeks. And at 8 weeks, we have 3 groups of mice: control mice, no antibiotics, mice continuing to receive antibiotics, or a group of mice that got 4 weeks of antibiotics and then we stopped. So now when we look at the circles, black circles are the controls, orange are continuing antibiotics. They're further apart than before, not surprising. One group is getting antibiotics. But here's the group that stopped the antibiotics. Their microbiota have reverted to normal. So the effect of the antibiotic was transient on the microbiota, but the effect on the phenotype was permanent. So this slide provides evidence that even a transient early life alteration in the microbiota can have long-term consequences because it affects a developmentally critical window. So we continued this work with an experiment that we called Transstat. We asked, could we transfer the growth phenotype using the microbiota alone to isolate that from the effect of the antibiotic? So the donors were mice that received antibiotics or not. We took mice in the middle part of each group, so we wouldn't bias things. The mice were sacrificed. We harvested their cecal contents, and we gave these via oral gavage to germ-free mice, mice that we had purchased in a bubble. Now we conventionalized these mice, and we housed them for the next 5 weeks under specific pathogen-free conditions so we could see what happened. So here's the study design again. Our donor mice received antibiotics or not. We harvested their microbiota. We gave them to the germ-free recipients who did not receive any antibiotics, and we followed them for 5 weeks. Here's the main results on morphometry: total mass, lean mass, fat mass. Black line is the control group. We found that the microbiota from the mice that had received antibiotics, the recipient mice, had increased total mass. No change in lean mass, increase in fat mass. So from this experiment, we have evidence that the microbiota alone is sufficient to transfer the metabolic signal. Now we're also interested in immunity. And this slide looks at expression of genes involved in intestinal defenses. And here we're looking at the donor mice. We're looking at 3 genes involved in the T helper 17 phenotype, genes involved in transcription and cytokines and antimicrobial peptides downstream of this gene. Here are the donor mice, the controls in black, the mice that received antibiotics in orange. We see this decrease. We've seen this across multiple experiments. And so now the question is, what happened when we transferred microbiota from these mice to the recipients? And in fact, in the recipients, we see the same pattern. So we have evidence that the microbiota are sufficient to confer the immunologic signal as well. Now, these are experiments based on the farm model, the model in which animals get antibiotics every day in their drinking water. That's STAT. But human children are getting antibiotics to treat their ear infections and their throat infections. They're getting pulses of antibiotics at very high levels. And so we created a second model, and this was work by Yael Noble when she was a student in the lab. Yael's hypothesis was that a series of short therapeutic dose pulses of antibiotics administered early in life will sufficiently change the gut microbiome to alter body composition. So in this experiment, mice got 3 courses of antibiotics, 3 pulses, either amoxicillin, the most widely used antibiotic in childhood, or tylosin, a macrolide, and that's the second most widely used class of antibiotics. They got one through their mom's milk while they were nursing, then 2 more doses, and then at a certain point they were put on a high-fat diet to bring out the phenotype. I want to show you the effects on their microbiome. Here we're looking at richness, the diversity. The blue line is the control group. Amoxicillin has some depression of diversity. The macrolide has severe depression of diversity, and they never recover. When we look at the Shannon index of evenness, we see the same phenomena. In this model, the antibiotics are having long-term effect, more than 100 days after the antibiotics have stopped. Now, Alex Alexienko, an informaticist in the group, did a Kolinsky analysis that showed that 4 groups was the best way to segregate these data with millions of sequences and 338 specimens. And here is a cluster analysis over time. Here's the timeline from the beginning of the experiment to the end. Each line here represents one group of mice. So here's the control mice. And they begin life in this green cluster. This is the mom cluster type of population. And when the high-fat diet begins, they all shift to this red cluster. The mice on amoxicillin look a lot like the controls, but we can see that there's some perturbation here. But the mice receiving the macrolide, they're perturbed from the very beginning, from the very first dose, and it only finally the strong selective effect of the high-fat diet brings them to the new normal. So the macrolide has totally disrupted their population. Now this is particularly interesting because a paper just appeared 2 days ago, a study from Finland looking at the intestinal microbiome and relating it to lifetime antibiotic use in Finnish children. And the point that was the most interesting to me is that there was a difference between macrolides and beta-lactams. So in this, and what they find is that 6 months after, if this is the control population, 6 months after macrolide, there's a big shift, loss of particular organisms, and it only very slowly comes back. And in this principal coordinates analysis, here are the control mice, and here are the mice on macrolide. Again, much greater disruption from the macrolide than the beta-lactam. Exactly what we found in the mice. A lot of consistency here. So how do we put this together? Our hypothesis is that antibiotics have an impact on long-term physiology through changes in the microbiota. Our idea of health is an ancestral microbiota where gut microbes are talking to stem cell populations. If they're mesenchymal stem cells, they have to make developmental decisions. How many times to go around, and do they become fat cells, muscle cells, or bone cells? And if they're immunocytes, what's their immunocyte maturation? With an ancient microbiota, we'll call this normal long-term physiology. But in an antibiotic-treated microbiota or some kind of deranged microbiota, the signals of the microbes to the stem cells are abnormal, creating abnormal early life development with later long-term physiology. This is our big hypothesis. Lots of people did the work I've talked about today. I've mentioned a few of them who've done the most important work. Il-Sung Cho setting up the STAT model, Laurie Cox, her work on the STAT, Shingo Yamanishi, gene expression in tissues, Yael Noble, work on PAT, Alex Alexienko, informatics. Many other people helped in all different aspects of statistics, mouse care, immunology, x-rays, sequence analysis, funding from many different sources, especially the US government, the NIH. Now those are studies in mice. What about people? So we have done 2 studies now in a cohort of children born in England in the early 1990s. The ELSPAC cohort, the Avon longitudinal study of parents and children. And we basically asked 2 questions. In the first study, we asked, is there a relationship between antibiotic exposure and early life body mass? And what we found is that antibiotic exposure during the first kids who were exposed in the first 6 months of life developed greater fattiness than the kids who didn't. This is now one of 6 studies that are showing data in the same direction. Early life antibiotics increased fattiness. We also asked, what about C-section? And we found kids born by C-section had increased fattiness when we controlled for all other known variables. There have now been 4 studies that have shown the same phenomenon as well. Turns out that obesity is not the only disease that's rising. There are many diseases that have been rising since World War II, since the introduction of antibiotics. Juvenile diabetes, doubling every 25 years. Reflux disease, asthma. The list goes on. So what do we need to do? We need to do research about the consequences of antibiotic use. And here I'll include C-sections and formula feeding and other antibacterials, things that disrupt the early life microbiota. And if we find that there are consequences, we have to educate the public and the profession about the risks of antibiotics. We need to develop narrow spectrum treatments, not broad spectrum treatments that have collateral damage, but ones that will treat specific infectious agents. That will require better diagnostics. We need to remediate. If somebody has to take an antibiotic, maybe we can replace the acutely lost microbes or enhance depleted ones with prebiotics. And maybe we need to reverse. We have to go to the Yanomami and archive those vanishing organisms and maybe replace them in the children of the future, and ultimately monitor, is it working or not? So I want to go back to the CDC studies of antibiotic use in the United States. And here we're looking at the cumulative data, as I showed you before. But shortly after this paper was published in the New England Journal of Medicine, a group from Sweden reported on their data. And what we can see is that at the age of 3, when our kids have gotten about 4 courses of antibiotics, the kids in Sweden have gotten 1.4 courses of antibiotics. And at 10, when our kids have gotten 10 courses, they've had 4 courses. So the Swedes are at least as healthy as we are. There's no epidemic of childhood mortality in Sweden. There's no epidemic of deafness or ear infections. It suggests that a priori we could reduce 60% of our antibiotic use without any problem. So my thinking about child health in the future is that the doctor, the pediatrician of the future is going to examine that baby and is also going to examine that baby's diaper and do an analysis and try to figure out which microbes is this baby missing, which global microbes, those are microbes that every baby needs, or which personal microbes that that particular baby needs. And the doctor of the future is going to administer them to optimize health and obviously assess to see if it's working. So I want to finish with an unabashed plug of my book. You can see that everyone is reading it. And also tell you that in New York, there's an exhibit at the American Museum of Natural History about the microbiome. It's a terrific exhibit, and I recommend it to everybody. And with that, I'm going to stop and see if anybody has any questions. Thank you for your attention. And I think that if you have a question, you should come up to the microphone.
Right here.
And there's the microphone.
Hello. First of all, thank you very much to you and everyone who's done all this research, because we can't make decisions without the data. So I love seeing all the data people are coming up with. One question I had, and you hit upon it a little bit at the end, but is if genetic fecal diversity really is less than before, is there a way to for it to come back, perhaps by ending unnecessary antimicrobial processes, or by, for lack of a better word, spreading the microbiome of everyone on the planet around to each other?
Yeah. Yeah. So if we can control antibiotic use better and minimize C-sections, except for the ones that are really necessary and things like that, then maybe the problem isn't going to get any worse. But how's it going to get better? If we have really lost all this diversity, it suggests that we're going to have to restore diversity, and that we will have to understand what are the key organisms. Again, these, what I call global organisms, the organism that every child needs for healthy development. And we're going to have to give them back. Just as today kids are getting vaccines every few months to protect them against important problems, maybe they're going to drink a solution of certain defined organisms, and that will repopulate their gut to optimize health. That's what I think. That's going to take some time. That's going to take a lot of science for us to figure out what are the right organisms. Yeah.
So I'm interested in your PAT-STAT study, and the difference is, why do you think there's a difference between the amoxicillin and the tylosin? Is it just the different mechanisms of actions of those antibiotics, or what's going on there? Do you have any ideas?
Yeah. So that's a wonderful question. I can just tell you that our results are quite consistent in multiple experiments, that the macrolide has a much greater effect on the microbiota. The simple answer is that it is a much broader spectrum antibiotic for the microbes in the gut. And it turns out that that's very important, because if you look at antibiotic use in childhood, beta-lactams are relatively flat, but macrolides are increasing dramatically, in part because they're so heavily marketed all over the world. So that's why I was so interested in this study in human kids, because it's the same.
Yeah, I haven't read it yet.
Yeah, it just came out 2 days ago. Right. But so if that's true, doctors will have to learn to be more selective about which antibiotics they're using. I don't know the reason, but I assume it's spectrum.
Macrolides are also newer antibiotics. They have not been around as long.
So the point in the front is that macrolides are newer. Actually, macrolides have been around for a long time, but erythromycin, which was developed 60 years ago, fell out of favor. And then in the early 1990s, two new antibiotics came along, clarithromycin and azithromycin. Azithromycin is the Z-Pak. Many of you are familiar with it. The Z-Pak has become the #1 used antibiotics in the United States. In the average year, there are more than 50 million courses of Z-Pak prescribed. That's one course for every 6 people in the United States.
That's fascinating. So I had a question about when you were talking about the gut microbiota and how it talks with stem cells, right? And they're releasing signals. So could you make a guess to what those signals are? Is there any future work that you're planning to sort of address that question?
Yeah, so we're working on that right now. That's a great question. And we're interested in, and remember in the Durastat experiment, the antibiotic exposure ended at 4 weeks, and we found the first evidence of adiposity at 12 weeks. So there's a window. Something is happening during that time. We're currently looking at epigenetic changes. We're also looking at the immune compartment as well, because a young animal can invest their calories in defense or mass. And if you don't have to spend as much on defense, you can build up. So we're interested in that hypothesis as well.
Hi. You mentioned several sources of antiseptic factors in terms of reducing bacteria. But what about prebiotic factors such as our diet, given that we're starting to realize how much our diet has shifted from extremely high fiber composition towards very low fiber composition, and more fats and proteins. And would that also be a problem if we were trying to introduce, you mentioned introducing other gut biome from donors, but would that even work if we didn't have the proper prebiotics in place?
Right. So that's a great question. We're not doing that much on diet. We're doing a little. We have evidence from our FAT-STAT experiment that diet does make a difference, that they're additive. We have evidence that the gut microbiota of mice on normal chow is richer than on a high-fat diet. And there's a beautiful paper published about 2 weeks ago by Justin Sonnenberg and colleagues from Stanford looking at the effect of a high-fiber diet or a low-fiber diet in mice, showing that on the low-fiber diet there was loss of diversity. And then they did 4 generations of transplantation and showed that the loss of diversity got worse in each generation. It's very consistent with our disappearing microbiota hypothesis. Antibiotics is not the whole story. It's part of this story. It's a very important actor, but there's no question that there's the possibility of synergy between different factors of modernization.
I have a question from Joe in Minnesota who's on the live stream. What are your feelings about our current food supply and our obsession with sterility in food and in our everyday environment?
Yeah. So there's several questions in there, and the first thing to say is that we have a general germophobia in our country, and very good marketeers and merchandisers are happy to take advantage of that and propel that. So there are so many products that are being sold trying to get rid of all the germs based on the theory that all germs are bad. And many people believe that. But certainly as microbiologists, we know that some germs are bad and some are very bad. Most germs are neutral to us, and some germs are actually good. And I wonder if by all the hand washing we're doing, where we're removing our own human microbiome, whether we're helping human health or we're hurting human health. We don't know the answer. About food and sterility, food in general is not sterile. It's never been sterile, but we want to make it safe. We want to avoid pathogens in food, and we've made great progress in doing that. It's not perfect, and so we have to understand. And one of the problems, of course, of the huge use of antibiotics on the farm is that we're getting antibiotic-resistant bacteria in our food. There are residual antibiotics in the food. How that's affecting us, we don't know.
If we're losing microbiomes by the generation, is there a point where humanity would not be able to bounce back and evolve? Could we break our genetics?
Mm-hmm. Right. Is there a point where we might not come back? And I'm sorry to say that that's chapter 15 in my book, which I call Antibiotic Winter, because we're talking about metabolism, but the real danger to humans is epidemic. And we know that the best protection against epidemics is healthy microbiota. That's been shown in many different experiments. So you'll have to read the book.
So could the data that you've put up here potentially be viewed as an indicator of health? You said earlier that an organism can spend its resources on either defense or mass. Would antibiotic use early in life that wipes away chronic low-grade pathogens that have been passed down for generations potentially be a good thing?
It could be a good thing. It could be a good thing. But the question is it a good thing? So theoretically it could be. But I guess I began this by thinking, why have all these epidemics started? Why has obesity skyrocketed? Why is juvenile diabetes doubling every 25 years? All in recent times. Allergies, peanut allergy, which was rare disease, celiac disease, inflammatory bowel disease, and more. And so the question is, what's changed? And you have 10 diseases, you could have 10 different causes. Or there's one factor that's fueling all of them, and they manifest differently in different individuals. So in one of the chapters of my book, I talk about height, about how people have gotten taller. And we generally think that that's a good thing. And we link that to microbes as well. You'll have to read the book.
I have another question from our live stream from Ruby. How do fecal transplants relate to what you've shown, and could they be used to reduce the damage to the microbiome?
Yes. So the question is, could fecal transplants reduce the damage to the microbiome? Right now, fecal transplants have been shown to be very useful for a very severe infection due to a pathogen called Clostridium difficile. This is a life-threatening pathogen. People may die from this infection. And the fecal transplant seems to be better than conventional therapy. So that proves the principle that transferring a normal microbiota can restore health. But most cases are not quite as severe as this, and fecal transplant is largely unexplored. This is a complex biological property. We knew with we were giving blood transfusions and we were transferring infectious agents. So this has to be very carefully controlled and studied. We really have to know what we're doing so that we don't get a new epidemic of hepatitis C or a new epidemic of HIV. as have happened when we've transferred human biological projects.
With the kind of decrease of many antibiotics and their effectiveness, and we're kind of seeing a continuing trend of more ineffective antibiotics and not a equal trend of new antibiotics to replace them, if you had to look forward 50 years and think about how that is going to be affecting all the research and all of the trends that you're seeing, that you've presented now, what kind of relationship do you see there?
Yeah, that's a wonderful question. There are several different answers to your question. The first is that one of the reasons that our antibiotics are losing efficacy against pathogens is that we're overusing them so much. Use selects for resistance. And so there are two sides of the overuse coin. One is the increase in resistance, and the other are these collateral effects on the microbiome that I'm talking about. So if we were to reduce use, we might slow down the spread of resistance. Now the next point is a little technical, but I'm going to try to explain it, and that is that when we talk about susceptibility or resistance, we're talking about the dose of antibiotic that will effectively kill a pathogen, something that is, we don't want that organism multiplying in our blood. We want a level of the antibiotic to suppress its growth so that our immune system can take over. And that's generally the doses for that, the levels are very high to achieve that inhibition. But none of these tests were really designed to examine the effect of the antibiotic on our normal microbiota. The fact that if you apply an antibiotic to a mixed culture of all these organisms, that there are going to be effects at low levels. The farmers proved it. They showed with stat, with very low doses of antibiotics, that there's a profound effect on growth. And the doses in stat, on average, are 1/50th the doses that we're using to treat our kids with ear infections. That's one of the big differences between stat and pad. It's dose, 50-fold.
We've got another question from our live stream. This one's from Galen. Are commercially available probiotic products, such as yogurt, Jamie Lee Curtis, all of that, are they good at replenishing the microbiome?
Yeah. So this is a question that comes up a lot. And in full disclosure, I eat yogurt just about every day for the last 40 or 50 years. I eat it because it's delicious and because I think it helps me, but I have no evidence. But if we've lost hundreds of microbial species, I don't understand how taking in 1 or 2 or 3 species would really help restore our diversity. And in fact, we know that if you take in antibiotics in yogurt today, you will find those bacteria, those live bacteria in your poop tomorrow, but not the next day, because they are immediately eliminated. The normal microbiota does not like to share its turf with outsiders. And most of the so-called probiotics on the market have not been tested in clinical trials, the rigorous clinical trials that we would need to understand whether they work or not. I think I only have time for about one more question, and then I have time to sign a few books. Okay?
All right. This one comes from Dusty on Twitter. Could the increase in obesity result from epigenetic changes that the microbe is communicating to the host cells?
Yes, yes. So that's a great question, and that's one of our leading hypotheses. We're trying to understand what's going on during that window from when the antibiotic stops and when we can see evidence of the obesity. And we are testing that hypothesis while we're here tonight. So I want to thank everybody for your attention and your great questions.
Thank you so much, Dr. Blaser. Thank you for everyone that sent in questions from online. And for our audience here, we will be adjourning to a reception out in the lobby. We hope that you all can join us. Dr. Blaser will be around for about maybe 10 or 15 minutes to be able to sign his book, and we do have those for purchase out in the lobby. So I hope you all will join us for a little more lively discussion outside. Thank you so much.
Once you're in the BSL-4 space, the only way you can get out is to go through a chemical shock.
It's an unusual room, never seen anything like this. Anybody who has access to this facility first has to go through an R scan. So the HEPA filters filter the air coming out of the facility, and that will remove bacteria, viruses, anything that might constitute any kind of risk, right? Remember, this building is basically a second building inside the main building.